Very promising treatments have arisen in the COVID fight, both on the treatment side and as well as on the vaccine side. Hello, everyone. This is Dr. Chris Martenson here with your SARS-CoV-2 daily update. I did take the last 2 days off. Really needed to, by the way. Took a whole weekend off. First one since I started this. And here with your honey badger virus, I'm calling this one Turn the Corner. And the reason for that is that as I look through the science of all of this, and again, this is who I am right here for those of you who are tuning in who don't know me or aren't familiar with me, there's some really interesting stuff out there in the data right now, and I think we've got a handle on what might be happening in a very promising treatment, of course, in theory, and we've been talking about this stuff for a while. But first, the news. Wow, I took 2 days off, and 272,000 more cases piled on, and this is yesterday's numbers so that we have the complete case count numbers. I'm recording this at a time where today's numbers for April 6th aren't in. So these are the April 5th cases. And Spain now in the number 2 position in the world. Just a really horrific death toll right here. Many thousands in the serious or critical condition. But notice that Italy now also, very horrific death count here. But turning the corner here for them on the amount of serious or critical cases. And also their case count is down. Remember, these used to be 7,000, 8,000. So they've flattened their curve, and they did that with a very aggressive level of NPIs, non-pharmaceutical interventions, social distancing, things like that. Germany still clocking in, with many more deaths than it had before, but very much fewer relative to other people. So let's just take a quick peek at this. Cutting all the way down the list here to the UK, has nearly 5,000 deaths here at 4,934. That's more than 3 times as many as Germany, but with only half the cases, less than half the cases. And so this is at least in part due to testing. Germany has a much more expansive testing program, but clearly something else is going on. We don't have all the answers for that yet. Is it treatment? Is it genetics? We don't have all the data on that yet. But certainly we have a numerator and a denominator problem. It's the numerator, how people are recording the deaths, and are all the deaths being recorded? There is news out that the United States has not been recording deaths properly, and I don't have the data for that in this update, but I've been reading those reports, perhaps you have as well, that this number might need to be twice as high. We don't know. So the United States now clocking in at over 1,000 deaths per day, and that's going to remain that way for probably a couple weeks at least, looking at the trajectory of all of this. And yeah, there's still a long way to go in this story, although I think maybe we've turned the corner a little bit. Remember, we did talks about chloroquine phosphate a long time ago. We'll get to that in just a second. But first, this is the most promising vaccine candidate that I've seen so far. This is a press release, came out on Thursday, comes out of University of Pittsburg. This is interesting, and I'll tell you why I like this one out of all of them that I've seen so far. And there's a whole wide variety of vaccine candidates out there ongoing right now. But this is the one that caught my eye, and here's why. So Pitt School of Medicine scientists announced a potential vaccine against SARS-CoV-2. When tested in mice, so only animal model so far, the vaccine delivered through a fingertip-sized patch produces antibodies specific to SARS-CoV-2 at quantities thought to be sufficient for neutralizing the virus, so that's a good first step. I haven't seen if these animals have been represented with the virus yet. I'd like to see that and see if that actually confers immunity for them. Don't know that yet. The paper published April 2nd in eBiomedicine, which is published by The Lancet, is the first study to be published after critique from fellow scientists at outside institutions that describes a candidate vaccine for COVID-19. So peer-reviewed. Good step. The researchers were able to act quickly because they had already laid the groundwork during earlier coronavirus epidemics. So they had some data, they had some research, they had some techniques that they could dust off. Continuing with this particular PR release here, let's look at this. Compared to the experimental mRNA vaccine, which is out there, that's the candidate that entered clinical trials. We showed you that. They had some healthy volunteers were given shots with the mRNA vaccine. That's a platform that is really new. Don't have a lot of data around how effective that can be. Seems really promising. This one is different. The vaccine described in this paper, which the authors are calling PittCoVacc, short for, I guess you can figure that one out, follows a more established approach using lab-made pieces of viral protein to build immunity And it's the same way that current flu shots work. So they're taking chunks of viral protein, putting them in your body. Your body says, hey, that's not natural, that's foreign to this body, and then it mounts an immunological response. So we know how that pathway works really well. The mRNA one it's got a pathway, I think, that's understood, but less well known how the body reacts to that one. This giving antigens, of course, is how most the vaccines work in the first place. So we know about those. The researchers also used a novel approach to deliver the drug called a microneedle array to increase potency. This array is a fingertip-sized patch of 400 tiny needles that delivers the spike protein pieces into the skin where the immune reaction is strongest. So, of course, they were targeting these spike protein pieces because the spike protein is the protein on the virus that binds to the ACE2 receptor and gains it entry into the cell. So, great place to target an antibody. So that's why you would give these pieces of antigen. The patch goes on like a Band-Aid, and then the needles, which are made entirely of sugar, and the protein pieces simply dissolve into the skin. We developed this to build on the original scratch method used to deliver the smallpox vaccine to the skin. And that's how smallpox was the very first vaccine. And the way that was delivered was cowpox, which is a related virus to the smallpox, but it's in a cow. So it doesn't really infect humans. But if you could take that same smallpox analog, in this case cowpox, and scratch it into the skin of a human, that would be enough for the human immune system to say, oh, look at this pox vaccine, this pox antigen, let me make antibodies against that. All right. But this is a high-tech version of that old method of scratching into the skin, and more efficient and reproducible patient to patient, actually pretty painless. Kind of feels like Velcro. Now let me tell you why I like this platform, if it works. Of course, it's in animals, it's got to go to humans, probably still about a year. But this, at least what you needed, and the things that were going to make me excited about this was, is this a pathway that we understand? Can we scale this up? So this is where it gets exciting. The system is also highly scalable. The protein pieces are manufactured by a cell factory, which are layers upon layers of cultured cells, engineered to express this spike protein. So this is done in vitro. You have certain cell lines that are then made to express this protein. So you can just do that in vast quantities. We know how to grow cells in quantities, so that feels good. This can scale up. That can be stacked further to multiply yield. Purifying the protein can also be done at industrial scale. Yes, mass producing the microneedle array involves spinning down the protein sugar mixture onto a mold using a centrifuge. Okay, that might be a little slow, but you can certainly build lots of centrifuges. Once manufactured, the vaccine can sit at room temperature until it's needed. That is a big deal right there. Because if it doesn't have to be kept refrigerated, and maybe, it's temperature dependent, so it's breaking down every time it's at room temperature, and then the efficacy is changing, all of that. So this idea that it can sit at room temperature, big deal. That eliminates the need for refrigeration during transport or storage. For most vaccines, you don't need to address scalability to begin with, Gamboto said, but when you try to develop a vaccine quickly against a pandemic, that's the first requirement. When tested in mice, PittCoVacc generated a surge of antibodies against SARS-CoV-2 within 2 weeks of the microneedle prick. Good, that's a good response right there. Those animals haven't been tracked long-term yet, but the researchers point out that mice who got their MERS-CoV vaccine produced a sufficient level of antibodies to neutralize the virus for at least a year. So that's against the MERS coronavirus, which is a close analog, but not exactly the same. So could be different, but that's a really good sign that it was able to show specificity and duration against the MERS antivirus, okay? So far, the antibody levels of the SARS-CoV-2 vaccinated animals seem to be following the same trend. Okay, so that's good. So they have data and they're comparing it, and so far, so good. Importantly, the SARS-CoV-2 microneedle array vaccine maintains its potency even after being thoroughly sterilized with gamma radiation, a key step toward making a product that's suitable for use in humans. That's fantastic. So it can be manufactured at scale. It confers an immunological response that mirrors one that we know is good, and you can sterilize it. So I like this. This is all really hopeful, and that's a really good sign. All right. Hydroxychloroquine, let's turn to this. We presented the chloroquine phosphate data, a month and a half ago, maybe 2 months ago now, when it came out of China. Now we're getting more data around it, and some of it's anecdotal. I'm still waiting for a big study. There's a lot we need to know about this to really use it well and effectively, but here's some really promising stuff. This was in New York Times. April 1st, the malaria drug hydroxychloroquine helped to speed the recovery of a small number of patients who were mildly ill from the coronavirus. Doctors in China reported this week cough, fever, and pneumonia went away faster, and the disease seemed less likely to turn severe in people who received hydroxychloroquine than in a comparison group not given the drug. So we actually have a control group here. A lot of people mistakenly have been telling me we can't do these sorts of tests because it has to be a double-blind control and it would be unethical. But here they actually put a control group in, and it didn't have to be blinded. They just figured out how to get a control group. So having a control, really important, so you can know what you're actually seeing. This is the first study I've seen that actually had that in it. There might have been others, but we'll go with this one for now. The authors of the report said the medication was promising, but that more niche research was needed to clarify how it might work in treating coronavirus disease and to determine the best way to use it. Yes, going to send a ripple of excitement out. It already has. We've seen that. Let's continue this article because I think there's good information in how the study was done that helps us understand how we might want to see it done and used if any of us had to use it. The new study of 62 patients with an average age of about 45 did have a control group. It was conducted at the Renmin Hospital of Wuhan University in Wuhan, China. The patients were carefully chosen to exclude people with medical problems that could be made worse by hydroxychloroquine, like abnormal heart rhythms, certain eye diseases, and liver or kidney problems. So this hydroxychloroquine does have some side effects that come with it. And one of them, it tends to lengthen the interval of the heartbeat, a portion of the heartbeat. And so people with abnormal heart rhythms, you wouldn't want to give them this stuff. And as well, I think it was wise to get people out who had liver or kidney problems. Half the subjects, the controls, received just the usual care given to coronavirus patients, and half had the usual care plus hydroxychloroquine. What I don't see here is if they use zinc. I think zinc is a big additive to this for a whole lot of reasons. But so this is just hydroxychloroquine compared against standard treatment. Plus you're getting the standard treatment with hydroxychloroquine. So the usual care included oxygen, antiviral drugs, antibiotics, and other treatments. So I don't know if that included zinc. I don't know if this includes Z-Pack, azithromycin. Don't know. Their disease was considered mild, even though all had pneumonia that showed up on CT scans. So that's still mild. Remember, mild is like you still have pneumonia. I wouldn't consider that mild, but here we are. After giving informed consent, they were assigned at random to either the hydroxychloroquine or the control group. They were treated for 5 days, and their fevers and coughing were monitored. They also had chest CT scans the day before the study treatment began and the day after it ended. Coughing and fever eased a day or so earlier in the patients who received hydroxychloroquine. So that's good. A day or so earlier is great. That would be very welcome. And pneumonia improved in 25 of 31, as opposed to 17 of 31 in the control. So 8 more people had improved pneumonia. But look, not all of them. 6 of them didn't here. And here we see that 14 didn't improve. The illness turned severe in 4 patients, all in the control group. 2 patients had minor side effects from hydroxychloroquine. 1 had a rash. Geez, I'd take that. And another had a headache. Yeah, that too. So you can see this clearly had a good effect. And again, I don't know if this was the full treatment that included zinc, but let's carry on. Here is a LA doctor who's trying this, seeing success with hydroxychloroquine. Dr. Anthony Cardillo has said he's seen very promising results when prescribing hydroxychloroquine in combination with zinc for the most severely ill COVID-19 patients. Every patient I've prescribed it to has been very, very ill, and within 8 to 12 hours, they were basically symptom-free. So clinically, I'm seeing a resolution. That's awesome. As well, I think there's a lot of data here that would suggest not only would we want to give this stuff to people who are severely ill, but we might want to give it to people early in the disease progression. So this is the use of hydroxychloroquine treatment by a Brazil virologist, got their PhD from Oxford, this Paulo here. Hydroxychloroquine was the Bolsonaro remedy and the Trump remedy, referring to the leaders of Brazil and the United States, respectively. Now they're under fire, including from within their own governments. Technically, the medicine should be given between the 2nd and 5th day of the disease. After that, the person needs to be hospitalized because they will need respiratory support, is a short therapy, and the adverse effects are not manifesting according to several studies. In Sao Paulo, the Prevent Network had 96 deaths from coronavirus as of March 22nd, almost half of all the deaths reported by the government of Sao Paulo. Today, they're with only one person in the ICU. Since Prevent adopted this protocol, there have been no more coronavirus deaths. And the people who had problems are those who entered late in this protocol, already with advanced disease. So I think the point I want to get to here is that this treatment works, and it works better if you give it early on in the treatment cycle. And that makes a lot of sense, right? So you wouldn't want to just wait until this progressed all the way till bilateral pneumonia and somebody's, just about ready to get intubated and they're like, last Hail Mary, let's try this hydroxychloroquine plus zinc. Nope, you want to try that early and often. So I think you need to start this early. The sooner the better. They're saying here between the 2nd and 5th day of the disease. Yep, that would probably be fine. Once you're on a ventilator, it's kind of too late. The stories I'm reading now say that once you go on a vent, you have about a 30% chance of coming off that vent alive. The odds are not good. So this whole idea that we need hundreds of thousands of ventilators may not turn out to be the case if we can prevent people from getting on them in the first place, which would be fantastic. Is it possible this could work prophylactically, meaning should healthcare workers be taking hydroxychloroquine before, as they go about their work? Maybe. We presented this several weeks ago that this is what India is doing. I don't have any follow-up data to suggest if that's working for them or not. But India is giving hydroxychloroquine prophylactically to their healthcare workers. And I got to point this out. It's not for everyone. There's plenty of comorbidities that would exclude its use. And remember, for most people, this is COVID is something that for most people is not. You would probably just want to get off it, survive it all on your own if you were having a mild case of it. To me, mild isn't by the time I have pneumonia. I would not want this to progress all the way to pneumonia. Interestingly, Sweden actually pulled the plug on hydroxychloroquine. It says chloroquine has been given to several hospitals in Sweden, but last week all the hospitals stopped the medicine. There were reports of suspected more serious side effects than we first thought. We cannot rule out serious side effects, especially from the heart. And it is a hard-dosed drug. I don't know what that means. This is a translation. In addition, we have no strong evidence that chloroquine has an effect at COVID-19, says the professor chief physician here at in Sweden. I don't know what they're up to in Sweden. This came from April 4th, by the way. But this is clearly showing very strong clinical promise in many, many places. And I didn't put up the most prominent case. Did I bring it up into this? Nope, I sure didn't. Which was a Dr. Zelenko in the United States, treating the Hasidic Jewish population up north of New York City. Has had tremendous success that he's been reporting out, and Trump quoted that doctor's results, and we're seeing lots of cases like that. The thing I haven't seen, which I'd love to see, is I want to see the data coming out of this, like we saw in that Chinese group where they said, okay, here's how we did it, 60 patients, broken into 2 groups. That's all good. I want to see more of that data because that's really going to help us understand how good this is. And again, without knowing what they did in the Chinese study that I quoted right here, we can see it's very good if it's improving that much. If you have 25 out of 31 improved instead of 17 out of 31, that's big. But again, this isn't 100% success. These people are reporting something closer to 100% success. Dr. Zelenko as well. So I don't know what to make of that yet at this particular point in time. As well, there was this thing in Sweden where they had this one anecdote where this guy, Carl, said, he had vision problems and, peripheral vision was reduced and all of that. So then he called the Poison Information Center, and it turned out the dose he was given was dangerous. So whoever was giving him, doctor had prescribed this, had given him too much of a dose. But that's just an anecdote. It turns out, today it no longer has any symptoms for COVID-19, right? Because he took the hydroxychloroquine. but thinks his vision is still worse than usual. He still feels a little dizzy, but he feels better than he did before. All right, I'm going to get into a novel theory about why hydroxychloroquine might work. It's very fascinating, but first, a lot of people have asked me about this ivermectin, right? Which is, again, it's another, it's basically a vet drug at this point in time. It's an antiparasitic. And at any rate, this has been sent around to me a bunch, and the antiparasitic drug available throughout the world, found to kill COVID-19 in the lab within 48 hours, shown a single dose of the drug ivermectin could stop SARS-CoV-2 and virus growing in cell culture. So that's the most important part here. Cell culture. Lots of things work in cell culture that don't work in the whole body. So this is interesting, but we're going to just put this out there in the interesting column for now. I would not recommend anybody, start chugging some ivermectin because you think you've got COVID at this point. There's a lot of work between cell culture and figuring out how this works in an actual whole body or a human. So here's this novel theory. This is an interesting one that came out, and I've been sort of struggling with it for a bit and trying to understand it. And anyway, it's interesting enough. I think I want to bring it forward now. So COVID-19 attacks the 1-beta chain of hemoglobin. And hemoglobin is that protein molecule that sits inside of every red blood cell and gives the red blood cell its color. It's red when it's bound up with oxygen. It's blue when it's not bound up with oxygen. So that's what hemoglobin is. It transports the oxygen around your body. And it says here that COVID-19 captures the porphyrin to inhibit human heme metabolism. So this is interesting because hydroxychloroquine is used, in its primary use is an anti-malarial drug. Malaria is a parasite that gets in your body and lives inside the red blood cells. And while it's inside the red blood cells, it's busy eating the hemoglobin and using the nutrients from those proteins to reproduce. So this is kind of interesting, and that there might be a connection here between COVID-19 and the blood. And so a lot of pieces snapped into place when I saw this come forward, and I just want to put it out there as an idea. The novel coronavirus COVID-19 is an infectious acute respiratory infection caused by the novel coronavirus. The virus is a positive strand RNA virus with high homology or similarity to bat coronavirus. In this study, conserved domain analysis, homology modeling, and molecular docking were used to compare the biological roles of certain proteins of the novel coronavirus. The results showed that ORF, ORF8, and surface glycoprotein could bind to the porphyrin, respectively. So these are two components of the COVID virus. So the COVID virus is this little package. It's got all these different proteins. We've heard about the spike proteins. That's one, but it has these transmembrane and surface proteins as well. So it's actually a collection of proteins and a lipid bilayer, and then some mRNA on the inside. At the same time, ORF-lab, ORF10, and ORF3A proteins, all part of the COVID virus again, could coordinate attack on the heme. I guess I'm going to put a word in there, on the heme, on the 1-beta chain of hemoglobin to dissociate the iron to form the porphyrin. So these proteins come in and they would come into the hemoglobin and they would then occupy a spot in there and displace the iron from its spot. And I'll show you a picture of that in just a second. The attack will cause less and less hemoglobin that can carry oxygen and carbon dioxide. So if that iron is displaced, it no longer can perform its function, which is to transport oxygen under one set of circumstances and then take and capture CO2 and transport that under a different set of circumstances. The lung cells have extremely intense poisoning and inflammatory due to the inability to exchange carbon dioxide and oxygen frequently, which eventually results in ground glass-like lung images. The mechanism also interfered with the normal heme anabolic pathway of the human body, is expected to result in human disease. According to the validation analysis of these finds, chloroquine could inhibit ORF-lab, or is that ORF1A? I think that's ORF1AB, ORF3A and ORF10 to attack the heme to form the porphyrin and inhibit the binding of ORF8 and surface glycoproteins to porphyrins to a certain extent. Effectively relieve the symptoms of respiratory distress. So what's being said here is that chloroquine actually could go in and inhibit these proteins, these virus proteins, from attacking the heme. All right, and if that happens, then this would actually have a positive outcome. So here's why. It turns out then that, when I asked the Dr. Rasmussen, how is it that the COVID virus, how is it that it actually attacks and kills you? Like what's the disease process? Because is it the virus coming in, replicating and bursting that cell? Is it because we have a cytokine storm attack because our bodies are attacking ourselves with an overmodulated immune response? Or what is it? Well, this is an alternative theory here that says that this is fundamentally a blood disease. So your blood cells, your red blood cells look like this. They've got this little funky shape. It's a membrane. And inside that membrane is just packed with these hemoglobin molecules. And each one of these hemoglobin molecules would look like this, got 4 big chains. And each one of these is carrying an iron molecule represented by these red dots. Oxygen would come down and bind to that. So the theory here is that these ORF proteins are coming in and displacing this iron and kicking that iron out. And here's the problem. That iron is going to be extremely toxic. It's going to have be either in a 2+ or a 3+ state, highly reactive compound, an ionic molecule that's going to it's really going to destroy stuff. So is this the issue? Is it that when those lung epithelial cells have all these virus particles in them coming out, and they've got these ORF proteins coming out of that? And if that's the case, then right there in the alveolar sac where you have these red blood cells attempting to exchange oxygen and carbon dioxide, what could be happening is those iron ions are getting kicked out, and they're very toxic. And they come out, and that's what's doing the damage in the alveolar spaces. All right, so that would sort of be the mechanism. You know what? This fits with the clinical data really, really well. First, what do we know? We know that one of the key signs is you see rapidly dropping blood oxygen levels. That would be consistent with blood that can no longer carry oxygen. We see bilateral ground glass opacities. It's always that bilateral. So one of the things that you see a lot in, say, bacterial pneumonia is it's not symmetrical. It's not bilateral. It'll be in the upper lobe or the left lobe and be the lower lobe and the right. It's somewhere. But here we're seeing it in both places. So that would either imply that the virus is just attacking the lungs with equal fervor in all places, or that this process of accidentally releasing the iron is happening in the bloodstream and or right there in the alveolar sac, so that would help to explain the bilateral nature of this whole thing. As well, one of the reasons people die is because multiple organ failure. Is that due to lack of oxygen, right? It's entirely possible that the multiple organ failure is due to that inability to carry oxygen as much as it is anything else. Maybe it's not due to the virus attacking the organs as much as we thought. It could just be simple old lack of oxygen. As well, lots of clinical data. I haven't presented it yet, but mega doses of vitamin C. That therapy really eases the worst of the symptoms, and it's been very, very helpful to people's clinical outcome to have mega doses of vitamin C. How does that work? Well, vitamin C is a very strong antioxidant. So as these Fe2+, maybe even Fe3+, very highly charged particles are, atoms and molecules are running around, vitamin C encounters those and neutralizes them. So this all begins to fit. Let me tell you, though, I have no clue how these COVID proteins would actually get inside the blood cells. I haven't heard a single mechanism that says that the virus binds and gets inside of a red blood cell. If it did, even if it did, it couldn't replicate in there because red blood cells don't have any of that machinery to do the viral replication process. So I haven't a clue what the mechanism or proposed mechanism might be for these ORF proteins to get liberated and to find their way inside red blood cells. But it's kind of interesting how this data is beginning to tie together as well. I finally have a place to hang my hat on this data, which we've noted a long time ago, but didn't have a way to explain it. It was just a curious little thing dangling in the wind out there, which was that blood type may affect COVID-19 risk. So a person's blood type may affect their risk for COVID-19. Curious, right? Researchers analyzed blood samples from nearly 2,200 COVID-19 patients in China, tens of thousands of healthy people, and found those with A blood types had significantly higher risk of COVID-19, while those with O blood types had significantly lower risk. So big puzzle. What's the puzzle about? Well, I don't know. If these ORF proteins need to get inside these blood cells somehow, if this is coated with type A antigens, maybe that happens more easily than if it's coated with type O antigens. Big mystery. Lots of stuff to get unpacked about that. But it's starting to look more and more like hydroxychloroquine fundamentally attacks the malaria parasite inside the red blood cell. Hydroxychloroquine is maybe doing something else inside this red blood cell that is then preventing this iron from being expelled out and doing all the damage. So that's the theory. Kind of interesting. Going to have to track that. But it begins to explain some of this. And I love having explanations that begin to make sense. So just a theory. Needs a lot of proof, but we'll see what happens. Remember, how do we beat this thing otherwise? Everybody wears a mask, and we have all these big reasons for that. And I want to just tell you, I love this story. I love this story right here. So how do you make a better homemade mask? This is the Sway Sew Shop story. I just love what these people did here. Lindsay Meadoff, the CEO of Sway Sew Shop, a 30-employee boutique in Los Angeles clothing manufacturer, wants the armies of people sewing homemade surgical masks to add a specific blue shop towel inside them. Madoff and two friends were appalled by the dozens of mask patterns calling for cotton, a highly breathable, permeable fabric, and became obsessed with finding a better fabric suited for the job. They built a homemade testing facility. These are my people right here, that could test particle filtration down to 0.3 microns, which is the N95 test, by the way, and tested every fabric they could find, from coffee filters to industrial materials. They discovered that by adding 2 blue shop towels and using a design that produces a tighter fitting mask, they could make a mask that could block up to 95% of the particles they could test, while cotton masks block 20 to 60% of the particles. This is brilliant stuff. Let's keep going with this story. I love this. Then one of these two people, Shempf, saw that the Centers for Disease Control and Prevention was telling healthcare workers to get proper gear, unable to get proper gear, to use a bandana as a last resort. I saw that too. I was like, a bandana? You got to be kidding me. The recommendation of a bandana made me ill. Me too, Shempf said. I couldn't understand how we can go from a 2020 N95 mask to a 1918 era cotton mask with a variable filtration of 20 to 60%. Still, a few weeks ago, before the CDC recommended cloth masks, Shempf, Madoff, and Madoff's business partner Heather Pavlou, a co-owner of the shop, became obsessed with finding a less permeable fabric for masks. We spent a few days researching and brainstorming any material that could filter: coffee filters, batting, window shades, Swiffer, interfacing, etc., all the way to more technical materials that are available to specialized industrial sectors like aviation, oil refinery, medical fields. Yeah, these are my people. I love how they did this. So they bought a $1,400 particulate counter device from Granger, that's a scientific company, that measures filtration ability down to 0.3 microns, and spent another 10 sleepless days testing all the fabrics they could. The ideal material turned out to be a stretchy blue shop towels made from a polyester hydro knit. Inserting 2 of these towels into an ordinary cotton mask brought filtration up to 93% of particles as small as 0.3 microns, the smallest their machine could test. Meanwhile, the cotton mask filtered 60% of particles at best. Polyester hydro knit towels are readily available at hardware and automotive stores. 2 brands they tested were Toolbox's Shop Towel, that's this one right here, and Zepp's Industrial Blue Towel. There you go, these right here. Interestingly, Scott's Pro Shop Towels, which were also made of a hydro knit fabric, didn't work as well. So this is just amazing. I love the science behind it. I love the testing. I love the ingenuity. I love that they were able to isolate differences between brands. They didn't just find one shop towel that worked. They tested a bunch of them. This is how it's done, people. I love this story. All right. And so as well, Pavlou said she sewed at least 15 types of the patterns that were being spread on the internet. So they weren't just comfortable with just the material itself. They wanted to find the best pattern. Before the team realized they were going to have to design a new mask themselves, so Pavlu tracked down and rented a port-account respirator fit tester 8040 machine. Yes. And the team tested things like how a wire nose clamp could help create a high filtration one-size-fits-all mask. And they are putting the final tweaks to their mask design and plan to release the design for free to the public next week. Their instructions will be good for home sewers and pros, available on Sway's website and its GoFundMe page. And for pros, meaning it's going to be an actual digital pattern that can be loaded into a machine that can cut fabrics and things like that. Hey, they have a GoFundMe. They're looking for money. I donated. I couldn't help it. I loved what this is. So if you're interested in what they're doing and helping to support actual citizens going out and taking the power back for themselves and doing the right things, this is it right here. That's the GoFundMe link. Want to see the other side of that story? So 2 days after this is released, we have the Surgeon General of the United States come out and, just in the most American thing ever, holds up a got naloxone t-shirt and then demonstrates how to fold it into an impromptu cotton face mask. This is the Surgeon General of the United States. First, what is the message? Naloxone is the antidote for an opioid overdose. So I'm not even sure what the symbolic meaning of holding up this particular t-shirt is, but that's what he went with. And as well, the idea that this is the best that we could do in a pinch as a nation. Didn't remember like in Apollo 11, and the astronauts need some help. And so they take all of these engineers who are just, on call at Houston. They say, here's what they have on the spaceship, go. And they throw a bunch of stuff on the table and these people have to work it out. Well, that's what these people did. They said, what do we have to work with? How do we work it out? And they went forward with it. But folding up a got naloxone t-shirt, come on. All right. That's just, I don't have words for that. That's real. It's embarrassing is what it is. It really is. And not up to the standards I expect from any civilized country, let alone my own. All right. Remember, plant a garden. Oh, hey, by the way, we have follow this link if you're wondering how to go about doing that, peakprosperity.com/DIY garden. And we've got an article up about how to go about creating a garden if you haven't done that before. Conclusions for today. Looks like hydroxychloroquine and zinc, it works. That's what the early data says, and seems very promising, and it looks like it's going to keep people off the ventilators. That's really critical. I think it has to be used as early as possible in the cycle. You don't want to give it only as a last resort. You give it more as a first resort. By the time that ground glass opacity has shown up in people's lungs, damage has been done. That is maybe possibly a chemical damage, if you will, from the released iron that's happening. We'll see. It's not right for everybody, this chloroquine, but it seems to be right for enough of people that it's going to be a really good frontline remedy. Is COVID-19 then actually a blood disease that liberates toxic iron in more than a cytokine storm disease? Well, that might be the case. So we're going to have to track that a little bit more. Just a theory at this point, but if true, it presents an entirely different clinical realm for going about treating it. And I got to say this, I'm just so damn proud of the Sway Sew Shop crew. Well done. That's how it's done. Data, science, ingenuity, some experimentation, data. Well done. And please, if you want to, people, go ahead and donate to those people at their GoFundMe so they can make lots of masks to go out to our frontline healthcare workers who still need them, promising treatments or no. That's all I have for you today. Thanks so much for listening. We'll be back again soon. Bye-bye.
Hi, folks. Adam Taggart here. Chris Martinson and I are the co-founders of Peak Prosperity. If you want to get alerted whenever we release a new video from Chris, just click the red subscribe button right beneath the YouTube video player. Once you've done that, a little bell icon will appear right next to it. Click on that bell. It looks like this. And that's it. The next time we publish a video from Chris, you'll immediately receive a notification from YouTube. Thanks for watching our videos.